So I think we could just get started. So on behalf of the organizing committee for the Young Scientists Program webinar series, I welcome you all for this fourth webinar. So this is for you that are just joining our webinar series. This is, comprises 10 webinars that will take place from between May and September this year. And this will be a kickoff event for the 20th IUPAB Congress that will be hosted together with the annual meeting from the Brazilian Society of Biophysics and the Brazilian Society of Biochemistry in October this year. So, in this webinar series, we'll have every 2 weeks, we have talks that will these seminars that will be include leading scientists in different areas of biochemistry and biophysics, as well as young scientists that are very productive and very active in these areas. And these young scientists, they have been selected among applicants from all over the world. So we have 32 selected participants from 14 different countries. So these meetings, they are taking place every 2 weeks, always at 2 p.m. Brazilian time, which this week we'll have well, before we get started to our webinar, I'd like to remind you that we have open applications for IUPAB fellowships. These are fellowships that will be were designed to cover the registration fee for participants to attend the virtual meeting that will be hosted in October. So the applications are open until June to the 30th. So I encourage all PhD students from that are attending these webinars or people that you know that would be interested to join. They can send their application until the end of June. And for further information, you can just visit the IUPAB website. There is displayed now here. So these fellowships, they are primarily for candidates from developing countries. So it's a way, to facilitate their participation in the meeting. So please apply and advertise for people that that would be interested in attending this meeting. So we now move to the program of our webinar, and I have to start with letting you know we're not that unfortunately the main speaker, Dr. Alan Dalton, he had a family emergency and just let us know that he's not going to be able to attend. So unfortunately, he has had to cancel. He had an issue, an emergency, and had to go to the hospital to see a family member. So unfortunately, we will not have his talk today. But I'm sure it's going to be a very interesting session. We have 4 talks from very talented young scientists from all over the world that will be speaking to us today. So I would like to invite now Khatereh khorsandi from the YARA Institute in ACECR, Iran. So she will have 15 minutes now to present her work. And then everyone will be able to participate with questions, either using the question and answer tab, or if you're willing to make a question orally, just raise the hand and the microphone will be open to you so you can make your question after her talk. So I'll just put her presentation on. And Khatereh, so please just say when you want me to move your slides forward, okay? Okay, can you see it?
Yes, I can see, but my video is not present.
I
mean,
your video.
Yes.
Yes, we can see you.
Yeah, everyone can see you.
No, really? Okay,
Yes.
because I cannot see myself.
Yeah, we
Okay,
can
that's
see
fine.
you.
Okay, I will. So I started now.
Yes, please.
Yes? Okay. Hello, everyone. First of all, I really like to thank the UPAB Young Scientist Committee for giving me this opportunity for have a talk in this great Young Scientist webinar series. My talk is about combination therapy of antimicrobial photodynamic inactivation and potential of nanoparticles and plant-based compound for using in APDT. So please, next slide. Hello.
Yes.
Next, please.
Well,
Next slide.
I did move. You're not.
No, I'm not seeing moving. Ah, yes,
No,
yes.
yeah, it moved. Yeah. We are now seeing the sunlight used for therapeutic agents.
Yes. Okay, as we know that the sunlight used as therapeutic agent for more than 2,000 years, like in Egyptian, Indian, and Chinese civilization, for many like diseases and problem like psoriasis, with vitiligo, skin cancer, and so on. So next slide, please. I don't know. I cannot see next slide.
Okay.
Is move?
I did move. Are you seeing the next slide?
Oh, yes. I see. Okay. I'm just here, like, want to talk brief about light mediated diagnostic. I mean, the using of light in therapy and also diagnostic. As we know that light mediated diagnostic under UV visible or near infrared light radiation that have different color and different energy and different penetration, as you can see in this slide, and depend on the application, we can use different wavelengths, like when we want to use it on surface, we can use it in lower energy, and when we need deeper penetration, we can use in red and infrared. So also they can apply in drug release or particle targeting for different application. I mean, in therapy or diagnostic. Okay, next slide, please. So in this slide, I want to say that the drug resistant is very important these days, and it says that the number of deaths caused by each year by pathogenic bacteria will increase to 10 million by 2050, and it will be a big issue in health. Around the world in health problem. So new biophysical substances or methods that could effectively cope with emerging drug resistance is necessary. So next, please. In this regard, antimicrobial photodynamic inactivation, or APDI or APDT, is one of the light-based treatment methods, such as laser-modified medicine and photodynamic therapy, that have been noted as having great potential against microbial infection. Antimicrobial inactivation, as I said, APDT, that also known as photodynamic antimicrobial therapy, is an alternative method to fight resistant resistant microorganisms, including bacteria, parasites, and virus, and so on. Next, please. So APDT method requires a presence of oxygen, and it's based on three important elements: presence of oxygen, non-toxic like dye that we call it photosynthesizer, and also light. And in the presence of these three elements lead to production of a singlet oxygen or free radical, which lead to bacterial destruction. Next slide. APDI mechanism is according to this fact that this compound, as I said, promotes oxidative stress, which result in DNA damage and destruction of cell envelope. Also, lipid and other compounds whose dysfunction of this macromolecule finally lead to cell death. As you can see here, it has two different mechanisms, type 1 or type 2. In type 1, we have radical production and destruction of the component I said, and in type 2 mainly is by singlet oxygen production, which lead to destruction of bacteria structure, lipid, and other components. Next. Application of APDT on treatment of superficial infection and the decontamination of surface and food also is another area for decontamination. Like it can use for decontamination in surface, in different surface, like in, for example, in dentistry, they use it usually. And also for decontamination of food, it can be used. Next. Actually, APDI has different positive effects. Like the main advantages of APDT is that the bacterial resistance doesn't develop as a result of photodynamics. It means that after PDT treatment or APDT treatment, the production of wildly active and indirectly target rust during APDT lead to destruction of bacteria, and they cannot take their resistance. The next one is APDT could affect the activity or production of nominal virulence factor, leading to decreasing bacterial pathogenicity. And this is unlike antibiotic because that antibiotic may cannot lead to decreasing the virulence factor, but APDT can. And another factor is APDI is not cytotoxic or phototoxic for eukaryote cells that are near to the area that maybe we want to have destruction of bacteria. So this method is not mutagenic effect on treated eukaryote or treated eukaryote cell around the prokaryote. Next. APDI has also more advantages than individual routine antibiotic, as you can see in this slide, like effective, as I said, it can affect both therapeutic window, like bacteria, parasite, virus, different, and also it can eradicate pathogen in biofilms, eliminate develop of resistance, destroy secreted virulence factor, as I said, and is practically safe for human tissue, not patient compliance, and also not leading to systematic that an antibiotic lead to systematic like side effect, and also APDT don't have this effect. And next. In this slide, the effect of elimination on bacteria cells has been shown as PS, like photosanitizer, that first is blue, active by light. It can lead to production of rust, as I said, like type 1 or type 2. And after production of rust inside the cell bacteria, and they can just by localize oxidative damage that created by rust, they can lead to destruction of bacteria like the cell wall, like inside like DNA, protein, and lipids, and then lead to destruction of whole structure of bacteria. Next, please. Another thing is that PDI can induce photo damage, like both morphological and functional changes. Morphological damage, like alternation of mesosome structure, direct damage to DNA structure, as I said, and functional damage could be like loss of enzymatic activity, protein oxidation, and formation of protein crusting. And all of these effects could lead at the end to the destruction of bacteria structure. Next. One important thing is that the difference in the cell wall of gram-positive and gram-negative bacteria could play an important role in, effectiveness of APDT on bacteria. Like gram-positive bacteria have a thick and porous peptidoglycan layer that surrounds the cytoplasmic membrane, so they are more penetrated in compared to gram-negative bacteria. But gram-negative bacteria passes the outer membrane surrounding a thinner peptidoglycan, and inside is a cytoplasmic membrane that here I just show for comparison both structure of gram-positive and negative. And next slide, please. For APDI or APDT, PS need to penetrate, as I just show in previous slide, need to penetrate or at least bind to cell wall of bacteria and end up in plasma membrane or cytoplasm, and then it should pass the bacterial cell limit as a simple diffusion and go inside the bacteria cytosol. So APDI of gram-positive bacteria seems definitely much easier to gram-negative bacteria, as they have a simple structure in compared to gram-negative. Next, please. One of the most important factors that lead to resistance of bacteria like Pseudomonas, like Staphylococcus aureus, is they can make the biofilm. And in many environments, bacteria exist as a complex and multi-species surface associated, like embedded self-produced matrix of extracellular chromatic substances composed of polysaccharide proteins, lipid, and extracellular DNA that is called biofilm. And bacteria that live in biofilm community possess several advantages for them, including structural stability, from adherent to biotic or abiotic surface, and increased virulence and resistance, as I said. And so, resistant to both antimicrobial and host immune response. So biofilm is one of the most factors that cause resistance of bacteria. And also biofilm is resistant to APDI. So in this regard, we need high PS and light dose also both for overcoming the resistance of biofilm. And in this regard, may block both the uptake of PS and the penetration of light, and thereby reduce the photosynthesizing efficacy. I mean that when we need more concentration of PS and light dose, so the biofilm in another way is resistant, so we cannot use more dose of PS and also light dose. Next, please. Also, there is possible mechanism of synergy between APDI and antibiotic. I mean that at the same time, when we can use APDI along with antibiotic, like they can have synergy by different mechanism, like mRNA inhibition, dysfunction of PB, DNA damage, and inhibition of synthesis of DNA. Modification of lipopolysaccharide, depolarization of membrane, alkalization of cytoplasm, and many different mechanisms that they can apply together for decreasing the resistance of bacteria. Next, please. Okay, as I said, APDI has three important parts, like light, photosynthesizer, and oxygen. Photosynthesizer could be different compounds that have like sharp absorbance in specific wavelengths, and after activation in that wavelength, they can go to higher energy and produce which lead to production of rust. Photosynthesizer could be curcumin like riboflavin, hypersine, methylene blue, and difference. And light source could be laser like LED. And next, please. According to the structure of a photosynthesizer, they have, as I said, and they have different absorbance in different areas. Like here, porphyrin has absorbance in like a red area. Phthalocyanine, like phthalocyanine could have maximum absorbance in like 700 nanometer, and different area according to their chemical structure. Next, please. So it depends on our work and that which photosynthesizer we could select for our work, and also light delivery is different, as I said, by lasers devices or LED, like nowadays fiber optic diffuser that they can use transdermally use or template grid with needles, wirelessly powered implantable LED light source, that they are very smart and also small, and can use for every part that we just need to use for destruction of bacteria. So next slide. Another issue in APDI is for infection, the best way to deliver a PS for APDT mechanism is locally, either by topical application. So the binding of PS to microbial cell, because it's rapid and mediated mainly by charge and charge interaction. So the best way for deliver is locally, and ideal PS designed for APDT should possess a wide range of characteristics like high quantum yields of singlet oxygen, and be a pure compound, a stable composition, soluble in water, selective to target tissue, safe and non-toxic for normal healthy. In this regard, next slide, please. There is, in this regard, there is some challenges in APDT, like most PS has hydrophobic characteristic, and so they penetrate in cell membrane, and under physiological condition they like to tend to form aggregate, so it just decrease their activity. And other challenge is they have low selectivity to their desired targets. Next slide, please. So for overcoming this challenge, nanomedicine is a good way, as a nanoparticle can act as direct carrier. As we know that nanomedicine is a particle-defined, a small object that have the size between 1 to 100 nanometer in size. They can use as direct carrier, like dissolve in matrix or absorb on the surface of nanoparticle. PS in this regard can put inside the nanoparticle or on the surface of nanoparticle, and in this regard, the delivery of photosynthesizer can happen perfectly to the desired site. Next, please. And for this application, nanoparticle can use in different way. Like I just presented here, as you can see, photosynthesizer could covalently bind to nanoparticle, including the dendrimer, as you can see, and or just like a lipid conjugated PS self-assemble into liposome or solid nanoparticle, or just biotargeting properties such as antibiotic. Another way is non-covalent encapsulation of PS, including nanomicelle, nanocapsule, nanosphere, and liposome, as you can see in another figure. Or there is nanoparticles themselves, for instance, a triodo, fullerene, or quantum dots that they are, and they can act themselves as photosynthesizer and capable of photodynamically like inactive microorganisms. Next slide, please. Example for PS contained inside the self-assembled nanoparticle. It could be liposome or micelle, that both of them are lipid-based nanoparticle. Liposome, as you can see in this slide, have a simple structure, controllable size, high load capacity, and flexibility for modification. And the PS can just put inside the liposome on the surface of liposome, or in hydrophobic, like hydrophobic drugs or PS can be on surface, or hydrophilic drugs can put inside the liposome. And a smaller liposome exhibit more efficient accumulation. And because the larger one can easily eliminate by macrophage or complement protein. Next, please. Another example for PS embedded in nanoparticle are polymeric or mesopore silica, as you can see in this slide. Silica nanoparticle prepared with well-controlled particle size. In the same time, several different PS can be encapsulated. They are non-toxic nature, inert and stable. They have potential non-reactive toward microbial and enzymes. So they are a very good option for delivery of photosynthesizer. Next, please. In this regard, we have done a research about, as you can see here, it's published in a PDPDT journal. It was about antimicrobial photodynamic and one-unit activity of curcumin encapsulated in silica nanoparticle. Next, please. In this paper and in this research, we use curcumin as a photosynthesizer, and we encapsulated curcumin because curcumin doesn't have good solubility. So silicon nanoparticle was used for increasing its solubility. And after synthesize this nanoparticle, curcumin silicon nanoparticle, as you can see here, we characterized it with different techniques. And after just characterization, next slide, please. We applied this nanoparticle that including curcumin as photosynthesizer against Staphylococcus aureus as an example of gram-positive, and also Pseudomonas aeruginosa. As you can see here, when we use curcumin silicon nanoparticle in compared to curcumin alone, it has a very good efficacy. So the planktonic form of bacteria in both group decrease significantly. Next, please. And also it was effective against the biofilm of both bacteria. So in another experiment, we applied this nanoparticle for finding that its efficacy against biofilm formation ability of both bacteria. So we saw that also bioformation ability of Pseudomonas and also Staphylococcus aureus decrease in nanoparticle form in compared to curcumin alone. Next, please. In the next step, we just want to examine its phototoxicity against human normal cells. So we selected human dermal fibroblast cell. And after treatment, we just want that it's that dose was effective for against bacteria. It doesn't have significant toxicity against human dermal fibroblast. Next, please. Another example of PS bonded, PS used with nanoparticle is PS could bind to the surface of nanoparticle. In this regard, We can example metal such as gold and silver, as you can see in this slide. Metal nanoparticle, they have a maximum absorption and scattering of near infrared, and also they can use for PDT of cancer. In contrast to silicon nanoparticle, metallic nanoparticle can be confined to extremely small particle size, so have very large surface and can just loaded with large dose of PS, as you can see in this slide. Next, please. In this regard, we just use a silver nanoparticle as an example of this group, and selected curcumin as a photosynthesizer, and we examined the effect of this nanoparticle against Pseudomonas aeruginosa because it's one of the important resistant bacteria. So we treated Pseudomonas bacteria with this nanoparticle, and we saw that after using the combination of nanoparticle and curcumin, the planktonic form of Pseudomonas decreased significantly in compared to AgE nanoparticle alone or curcumin alone. And next, please. Also, we examined this nanoparticle against biofilm formation ability, and we just find that the bioformation ability of Pseudomonas also decreasing by increasing the concentration of combination of AgE nanoparticle and curcumin in compared to AgE alone or curcumin alone. Next, please. In another experiment, we used this combination for destruction of biofilm, and we saw that also the destruction of biofilm was decreased, but this method was more effective in decreasing the planktonic form and decreasing the bioformation ability in compared to destruction of biofilm. Next slide. And also we wanted to know the cytotoxicity or phototoxicity of this combination, like AgE nanoparticle and curcumin against human normal cell. And we use it, examine its effect against human dermal fibroblasts. And as you can see here, it wasn't toxic against human dermal fibroblasts in the concentration that was effective against Pseudomonas aeruginosa. Next, please. In this slide, I want to talk about the mechanism that nanoparticle could damage bacteria. Nanoparticle, as you can see, could accumulate on the membrane surface and locally deform lipid or lipid structure. And after, damaging the membrane integrity, the photosynthesizer may accumulate on the cell membrane or release in extracellular. In another way, photosynthesizer could diffuse through the cell membrane, or also could just by endocytosis forming the endosome and releasing nanoparticle and allowing photosynthesis to localize intercellularly. So with different mechanism, nanoparticle that carry the PS can enter the bacteria cell and affect that its structure. Next, please. Last part of
Khatereh, you have just 2 minutes or 3, okay? Please.
Okay. So just I'm telling that with a nanoparticle themselves was another part of PS. So next, please. The example of this group could be that a photosynthesizer molecule that can be used in this regard, like fullerene, like graphene oxide nanosheet, TiO2, and different. Next, please. In this slide, I just show that fullerene as a group of carbon nanoparticle. And next slide, please. I'm just pass it quickly to say that my other research and TI order. Next, please. I'm not telling that, explain that. Quantum dot is another, just one. Okay, and next, another example is upconversion nanoparticle. So next, okay, next slide. I want to talk about this research that is about combination therapy of APDT with a phenolic compound. That in this experiment we use routine as flavonoid compound and photodynamic inactivation and using it like combination with APDT. Next slide, please. And after combination of routine as flavonoid compound as plant-based compound, we saw that by combination of routine with APDT, we have more efficacy against plankton form. Also, next slide, biofilm form of bacteria. And next, also we examined it against human dermatofibroblasts, and at that that concentration, it was a non-toxic. And next slide, please. And the mechanism that we find that how routine can affect the methylene as photosynthesizer, it was that routine can just stable the monomer of methylene blue, because in the solution methylene blue exists as monomer and dimer. And dimer form of methylene blue has low efficacy, but for us it's important that methylene blue stay in monomer form. So routine can just lead to interact with methylene blue and stabilize the monomer of methylene blue, as we just find it with the UV spectroscopy. And next is the last one. As conclusion, I want to say that nanoparticle has a different application, and we can use it for increasing the efficacy of APDT against different bacteria, fungi, or also for tumor cell. And also using plant-based compound could be another strategy for increasing the APDT efficacy. So thank you so much for your consideration. And I think because of my presentation, you just present my slide, it takes time for saying next. And I just it got, more time.
Thank you.
We have questions, but I don't know if we have time, or we can leave it to the end.
Maybe one question or two, and then we move forward.
Yeah. Okay. There's one from Dr. Watts. Have you explored the use of lipid nanodiscs, which have a high uptake capacity for a wide range of drugs and molecules, in particular the phobic ones?
Where can I see the question? Because
Sorry?
I couldn't hear you. I mean that they write the question.
Yeah, in the question and answers. You can see
In
it's
the
the third one.
answer. The third one. Explored lipid nanodiscs, which has a high capacity. No, we didn't use yet a lipid nanodisc for as an example of nanoparticle. In particular, hydrophobic molecular compound are more stable than liposome. They have been used for directing. No, yet I don't have experience with lipid nanodisc, but like for a nanoliposome or other lipid-based nanoparticle, we use, also other researchers use curcumin or other fields like methylene blue for increasing the efficacy of APDT.
Mm-hmm.
Okay. So thank you, Khatereh.